specifically infectious disease. The collection of cells, tissues, and molecules that mediate resistance to infections is called the immune system, and the coordinated reaction of these cells and molecules to infectious microbes is the immune response. Immunology is the study of the immune system and its responses to invading pathogens. The importance of the immune system for health is dramatically illustrated by the frequent observation that individuals with defective immune response are susceptible to serious, often life-threatening infection. The impact of immunology goes beyond infectious disease. The immune response is the major barrier to successful organ transplantation and increasingly used therapy for organ failure. Attempts to treat cancers by stimulating immune response against cancer cells are being tried for many human malignancies. Abnormal immune response are the causes of many diseases with serious morbidity and mortality. Host defense mechanism consists of innate immunity, which mediates the initial protection against infections, adaptive immunity, which develops more slowly and mediated the later, even more effective defense against infections. Adaptive immunity. Antigen recognition is the adaptive immune system. Antigen recognition in the adaptive immunity. Antigen recognition in the adaptive immune system. Antigen recognition in the adaptive immunity. Adaptive immune response are specific for the antigens that initiate these responses because the activation of lymphocyte is triggered by recognition of the antigens. Specific antigen recognition is the task of two structurally similar types of glycoprotein: membrane-bound antibodies on the B cells, membrane-bound antibodies on B cells, T cell receptors on the T lymphocytes. Cellular receptor in the immune system serve two functions. Cellular receptors in the immune system serve two functions: detection external stimuli, antigen for the adaptive immune system, triggering responses of the cell on which the receptor are expressed. These features of lymphocyte recognition and the antigen receptor raise two important questions. How do the antigen receptor of lymphocytes recognize extremely diverse antigens and transmit quite conserved activating signal to the cell? How do the antigen receptor of lymphocytes recognize extremely diverse antigens and transmit quite conserved activating signals to the cells? How is the vast diversity of receptor structures generated in lymphocyte? How is the vast diversity of receptor structures generated in lymphocyte? How is the vast diversity of receptor structures generated in lymphocyte? Antigen receptors of lymphocyte. The antigen receptors of B and T lymphocyte have several features that are important for the function of the receptors. These receptors recognize chemically different structures, consist of region or domain variable. V regions vary between clones of lymphocyte for antigen recognition. Constant C regions conserved among all clones of structure and integrity or for effector functions. Within the V regions, much of the sequence variable is concentrated within the short stretches, which are called hypervariable region or complementarity determining region. Complementarity determining regions, CDRS. CDRS, the parts of the receptor that bind antigen. They are complementary to the shape of the antigen, maximize the variability while retaining the basic structure of the receptor by concentrating sequence variation in small region of the receptor. There are special genetic mechanisms for introducing variation in the antigen recognition, recognizing region of this receptor while using a limited set of genes to code for most of the receptor polypeptide. antigen receptor are non-covalent attached to other variant molecule. Those function is to deliver the to the inside of the cell the activation signals that are triggered by antigen recognition. This is the T cell receptor, antibody immunoglobulin, signal transduction, major histocompatibility MHC. This is the antigen, the TCR, CD3, T cell receptor, antibody immunoglobulin and the T cell receptor. From antigen recognition, macromolecular protein, polysaccharide, lipid, nucleic acid, small chemical conformation, and linear epitopes. Here in T cell receptor peptide displayed by the major histocompatibility molecule in antigen presenting cells. This is linear epitopes. Diversity. Each clone has a unique specificity, potentially for 10 raised to power 9 distinct specificities. Each clone has a unique specificity, potentially for more than 10 raised to 11 power 11 distinct specificity. Antigen recognition is mediated by variable V region of heavy and light chain of membrane Ig, variable V region of the alpha and beta chain. In T cell receptor, this comparison between antibody and the T cell receptor. Signaling functions are mediated by protein Ig alpha, Ig beta associated with membrane Ig. Proteins. The signaling functions are mediated by protein CD3 and associated with other proteins associated with T cell receptor. Effector function are mediated by constant C region of the secreted Ig. And in T cell receptor doesn't serve effector functions. The collection of antigen receptor and the signaling molecule in B lymphocyte is called the B cell receptor complex. B cell receptor complex, BCR. In T lymphocyte, it's called T cell receptor complex, TCR. When adjacent antigen receptor of lymphocyte bind to two or more antigen molecules, the receptors are pulled together in an aggregate. This process is called cross-linking. It brings the associated signaling protein of the receptor complex into close proximity. When this happens, enzymes attached to the cytoplasmic portions of the signaling protein catalyze the phosphorylation of other protein. Phosphorylation trigger complex signaling cascade that culminate in the production of numerous molecules that mediate the response of the lymphocyte. Phosphorylation. Antibodies, immunoglobulin Ig may be membrane-bound antigen receptor of B cell or secreted protein. TCR exists only as a membrane receptor. Antibodies. An antibody molecule is composed of two identical heavy chain and two identical light chains, with each chain containing one variable region and one or more constant regions. Each light chain is attached to one heavy chain, and the two heavy chains are attached to each other all by disulfide bond. A light chain is made up of one V and one C domain, and the heavy chain has V and 3 or 4 C domain. Each domain folds into a characteristic three-dimensional shape, which is called the immunoglobulin Ig domain. This is the structure of immunoglobulin: secreted Ig, heavy chain, light chain, and this Fc receptor, complement binding site, and this Fc region, FAP region, membrane IgM, antigen binding site, plasma membrane of B cells. crystal structure H chain in red blue, in red and blue, L chain in green, crystallography. Each variable region of the heavy chain called VH or of the light chain called VL contain 3 hypervariable region or CDR. CDR3 is also a portion of the Ig molecules that contribute most to antigen binding. The fragment of the antibody that contains a whole light chain with its single V and C region attached to the V and the first C region of the heavy chain containing the portion of the antibody required for antigen recognition and is therefore called FAB, fragment antigen binding FAB. The remaining heavy chain C region make up the FC regions. In each Ig molecule, there are 2 identical FAB regions that bind antigen and one FC region that is responsible for most of the biological activity and effector functions of the antibodies. Between the FAB and the FC region of most antibody molecule is flexible portion called the hinge region. The C terminal end of the heavy chain may be anchored in the plasma membrane as seen in B cell receptor, or it may terminate in a tail piece that lacks the membrane anchor so that the antibody is produced as a secreted protein. There are 2 types of light chain called kappa and lambda, which differ in their C region but don't differ in function. There are 5 types of heavy chains called mu, epsilon, which also differ in their C regions. Antibodies that contain different heavy chains are said to belong to different isotype or classes and are named according to their heavy chains. Example: IgM, IgD, IgG, IgE, IgA. Regardless of the light chain class, IgA subtypes, H chain, serum concentration, serum half-life, secreted from, and the function. The comparison between the isotypes of antibody IgA, IgD, IgE, IgG, IgM. There is subtypes IgA 1, 2. In IgD there is no subtypes. In IgG, G1, G4, G1, 2, 3, 4. IgM none. Serum half-life per days, serum concentration milligram per deciliter, and the membrane immunity, mucosal immunity, neonatal passive immunity. IgE, the function mucosal immunity or nasal passive immunity. In IgD, naive B cells antigen receptor. IgE, immediate hypersensitivity, so it has a role in immediate hypersensitivity reaction. IgM has a role in opsonization, complement activation, antibody dependent cell mediated cytotoxicity, neonatal immunity, feedback antibody of B cells. IgM, naive B cell antigen receptor and complement activation. Antibodies are capable of binding a wide variety of antigen, including macromolecule and small chemical, because the antigen binding region of the antibody molecule forms a flat surface capable of accommodating many different shapes. The parts of antigen that are recognized by antibodies are called epitopes or determinant. The recognition of the different antigenic determinant may depend on sequence, linear determinant, or shape conformational determinant. The strength with which one antigen binding surface of an antibody binds to one epitope of an antigen is called affinity. Each Ig, IgD, IgE antibody molecule has 2 antigen binding sites secreted. IgA is a dimer and therefore has 4 antigen binding sites, and secret IgM is a pentamer which 10 antigen binding site. Therefore, each antibody molecule can bind 2 to 10 epitopes of an antigen as long as identical epitopes are present sufficiently close together. T cell receptor TCR. The T cell receptor for a peptide antigen displayed by major histocompatibility complex MHC molecule is a heterodimer composed of an A chain and beta chain, alpha chain and beta chain, each chain containing one variable region and one constant region. The structure of T cell receptor and this crystallography on the right, V chain, alpha chain and beta chain, and this transmembrane region, disulfide bonds. In the V region of the each T cell chain there are three hypervariable or complementarity determining regions. As in antibodies, CDR3 is the most variable among different T cell receptors. Unlike antibodies, both T cell receptor chains are anchored in the plasma membrane, and these cell receptors are not produced in the secreted form. Also, T cell receptors don't undergo class switching or affinity maturation during the life of T cell clone. Both the alpha chain and the beta chain of the T cell receptor anticipate in specific recognition of the major histocompatibility molecule and bound peptide. 10% of the T cell in the body express a receptor composed of Y and epsilon chain, which is similar to the alpha beta T cell receptor but has a very different specificity. T cell recognizes microbes that commonly contoured at epithelial surface, but either the specificity nor the function of these T cell is well established. The T cell receptor recognizes antigen, but it's incapable of transmitting signal to the T cell. Associated with the T cell receptor is a complex of protein called CD3 molecule and the chain that make up the T cell complex. T cell activation requires engagement of the co-receptor molecule CD4, CD8, which recognize polymorphic portions of the MHC molecules. Antibody immunoglobulin, T cell receptor, antigen presenting cells, major histocompatibility antigen, signal transduction, the Ig antigen and Ig beta and alpha, and this is a signal transduction. The difference between antigen receptor of B and the T lymphocyte, the antigen binding region, structure of antigen recognized, and affinity of antigen binding, one rate or of rate, accessory molecule involved in the binding, immunoglobulin IgG antigen, made up of three CDRs, linear and conformational. Structure of the antigen recognition in Ig of the macromolecule and small chemical, only 1 to 3 amino acid residue of a peptide and the polymorphic residue of on MHC molecules, affinity of antigen binding, average affinity of antigen anti-Ig increased during immune response. Development of immune repertoires, how the enormous diversity of these receptors is produced. There are many clones of lymphocyte with distinct specificity, perhaps as many as 10 raised to power 9. It's previously unreasonable that very possible receptors were encoded by one gene. A large fraction of the genome would be devoted to coding for antigen receptor only. In fact, the immune system has developed mechanisms for generating extremely diverse repertoires of B and T lymphocyte, and the generation of diverse receptors is intimately linked to the process of lymphocyte maturation. Maturation of lymphocyte. The maturation of lymphocyte from bone marrow stem cell consists of three types of process: proliferation of immature cells, expression of antigen receptor antigen genes, expression